33rd Books and Bagels program. So we welcome you here. If it's your first time, welcome. The way Books and Bagels works, it's going to be, we have 2 graduate students today. We're going to talk a little bit about their research and kind of what drives them and what motivates them in their research. Then we're going to have, at the end, some time for questions, as well as some discussion between our speakers. And our fill-in moderator today will be Dr. Tek Halim from Graduate Studies. Unfortunately, our moderator from the College of Medicine could not join us today last minute, so we will have Dr. Lim fill in, and he's glad to be here. So let's get started. Our first speaker is John K. Lee. He's an MD, PhD candidate in neurobiology and anatomy, neuroengineering, and bioengineering. He is from the Drexel University College of Medicine and School of Biomed, so he's a dual degree candidate with us. And he's going to talk today about how to win a Nobel Prize and other lessons from the 64th Lindau Nobel Laureate meeting, which he just attended. And John, you can start.
Okay, so I guess I'll be going against what most people talk about, which is their research. I'm talking about an opportunity I had to attend the Lindau Nobel Laureate meeting this past summer. So just to give you a little brief overview, the title is a little facetious, but I think that the lessons that I'm going to present are the things that I learned while at this meeting can really apply to all graduate students. I know it certainly applies to me. So we all know about the Nobel Prize, and we know that you can win them in lots of different categories. But just to begin, the Nobel Prize was created by Alfred P. Nobel, who was a chemist, engineer, inventor in the 19th century. And we know him for the Nobel Prize, but in his time, his contemporaries regarded him as a great inventor, in particular of dynamite. And he was a very successful businessman. And the story goes that a couple years before his death, he wasn't sick or anything, but a couple years before his death, a French newspaper accidentally released an obituary of him and called him the Merchant of Death. So he was able to figure out what his legacy was before he actually died. And he was a little disturbed by this, in fact, very disturbed by this. And so in 1897, when he died, he left his vast wealth and fortune for the establishment of Nobel Prizes to be conferred upon those who, for the betterment of humanity. And he established them in 5 categories: physics, chemistry, physiology of medicine, literature, and the Peace Prize. And then in 1950, in 1901, the first Nobel Prizes were given. And in 1951, 2 German physicians in southern Germany, in Bavaria, from this island of Lindau, they asked the Swedish count, who was living in Germany at the time, to create an annual meeting where Nobel Prize winners could meet with young German scientists to talk about research, to collaborate, and to get inspired. And what ended up happening was that over the years, these meetings expanded, and they went to covering all of the Nobel Prizes, except for, I believe, the Peace and Literature Prize. So in the 65 years since, there have been meetings on every year. There have been meetings switching between the various disciplines: economics, physics, chemistry, and physiology or medicine. And I believe next year there will be a meeting for all 4 disciplines. Whereas this year there was a meeting for physiology or medicine in June, and in August there was a meeting for economics. So the meeting is held in this island, Lindau, located on Lake Constance in the Bodensee in southwest southern Germany. And this is actually a very tiny island. From the train station over here to the hotel/Inselhalle where we were, it's about a 12-minute walk. So it's not a very big island, but it's every summer it gets filled up full of students and Nobel laureates. So here's some pictures I just want to show you. This is the Inselhalle, where they have all of the lectures, the opening ceremony, and where everyone gets to meet and greet. This is just a picture of one of the main squares of the island. This was taken on a very nice day. The first day I was there was actually pretty gloomy, but it's still, a very nice little German town. And I was there during the World Cup, so this is kind of the quietest it got. It got very loud and raucous later on in the week. Okay, so this year's meeting, there are about 37 Nobel laureates who were invited. I believe actually only 35 attended. And there were more than 600 young scientists from almost 80 countries. I believe in the US there were about 50, I want to say 50 students that were invited to attend. And the topics this year covered subjects such as cancer, immunology, AIDS, optical microscopy, and future research approaches in medicine. And while these are topics that related to the science of the meeting, the actual purpose of the meeting was to get young scientists to talk to each other and to meet with people who were considered very successful in their field, to get inspired, to educate one another, and really to connect with each other. And here's a list of the Nobel laureates that attended this year. It's small font, but I'm sure some of you recognize these names. I'll talk a little bit about a few of them later on in the talk, but I'm sure some of you have even come across their papers in your research, and they've inspired you to do the research you're doing right now. And the way the day-to-day schedule was arranged was that in the morning there would be plenary lectures given by the Nobel laureates. So 30-minute lectures given by the laureates, followed by seminars and then smaller discussion groups run by the Nobel laureates with young researchers in attendance. Evenings were filled with academic dinners sponsored by various funding organizations. We had one Bavarian night where we were taught how to do traditional Bavarian dances. And I think the country of Australia sponsored another evening where we all got to eat kangaroo meat, which is pretty nice. And then interspersed throughout the week, there were master classes that were run by Nobel laureates. And it was really an opportunity for graduate students to present their research to Nobel laureates and get feedback, but also to present to other people. I had an opportunity to attend one, and it was pretty inspiring. I would say 2 to 3 hours talking with people from various different fields about their research. And here's just a picture of what the lectures look like in the morning, and then the smaller discussion groups in the afternoon. I think you can see my arm. Yeah. And I'll talk more about this later. And this is the picture of the harbor at Lindau. So throughout this week, I was sitting there and meeting lots of people, having a lot of fun. And I noticed that there were sort of 7 key points that kept on coming up over and over and over again. And what I wanted to do in this talk is to sort of present those 7 points. And the title is facetious in the sense that it's not really how to win a Nobel Prize, but I think what it really talks to about is how to be a successful graduate student, and maybe even to be successful in other fields outside of science, outside of academia, just sort of in your everyday life. But these are 7 lessons that I think these Nobel laureates exemplified, and I think we could all learn from. I know I could definitely learn from their example. So number 1, believe in your research, and subtext to that is don't be afraid to test your hypotheses. Now we all believe in some form or another that our research is correct. The work we're doing is the work that we should be pursuing. But I really want to hammer this point in using this example of Dr. Barry Marshall. He was an Australian physician who won the Nobel Prize in Physiology or Medicine in 2005, and his study was on Helicobacter, or bacterium that grows in the stomach, or can grow in the stomach, and its role in disease. Up until the 1980s, it was believed that particular stomach problems like ulcers were created just by stress. So if a person had an ulcer or ulcer problems, peptic ulcer disease, it was just stress. That person was fine otherwise, but they just needed to calm down. They smoked too many cigarettes maybe, or maybe they drank too much alcohol, but they really needed to just not have stress. And his work actually found that this bacteria might be responsible for it. And in the 80s, he came across a lot of backlash. There was a lot of inertia that he had to overcome in the field. Because he said this in a talk in 1998, everyone was against me, but I knew I was right. And his talk really talked about, his lecture was really about the false illusions of knowledge that are barriers to innovation. I'm sure we've all come across this in our lives where people are telling us that we're wrong, or that what we think to be correct is not true. And I think his example, and this is a picture taken from the actual day, can be fairly inspiring to the rest of us. So this is a picture of Helicobacter and a stain in the stomach, and you can see these little black dots are actual bacteria. And up until then, people did not believe that bacteria could grow in the stomach, right? Our stomachs are full of acid, pH of 2, nothing could ever live there. And so in his research, he thought, and he believed with full conviction that this was the cause of disease in the stomach. So he did what any insane graduate student would do. He decided to test it on himself to prove his point. At the time in Australia, he said it was very difficult to get approval to do human studies, and his studies on pigs weren't working. And so he said it was better to ask for forgiveness later than to ask for permission. And so this is a picture of a cartoon that was released all throughout Australia, and it's actually him drinking the Helicobacter. He developed gastritis, and he developed some other health issues, and it really proved a point that Helicobacter 1 could grow in the stomach, and 2 could be the cause of disease. So I think the lesson for all of us, and I'm sure there are many of us here who work with very different types of diseases potentially, or different applications where we wouldn't necessarily want to test it on ourselves. I work with spinal cord injury, and let's just say I wouldn't do that to myself. So I think the issue though is that you really have to believe in what you're studying, to the point that you are willing to test that hypothesis. Maybe not on yourself, but willing to test it to the extreme. Okay, so the second point is that you will fail. And every Nobel laureate that came up hammered this point home. Failure is not an option because failure is inevitable. So the key point here is to fail fast. And this was said several times throughout the conference as well. And I'd like to bring up Stephen Chu, who won for the Nobel Prize in 1997 in physics for cooling and trapping of atoms and manipulating their movement with lasers. And you might also recognize him as our former U.S. Secretary of Energy under President Obama's first administration. And in his presentation, he talked a lot about failure as an inevitability, but an integral part of science. And he said the important part of failure is to learn to fail fast. Everyone will fail. He himself said that in his graduate school studies, he bounced around from lab to lab 3 or 4 times just because his experiments weren't working. But what he learned to do during that time was to not procrastinate, not take those failures to heart necessarily as a failure on his part, but a failure of just his inability to learn as quickly as he could. So he decided that if he was going to fail, he would fail in an hour, he would fail in a week, he would fail in a month, but after that, he would move on, and he would fail fast. And here are just some images. His talk on optical microscopy was actually was an excellent talk, and I think a lot of people who were in biomed just didn't understand the physics of it, but I enjoyed it very much. And I was able to attend a breakfast that he led a discussion in as well. Okay, so the third point, don't be afraid to try something different because you might end up winning a Nobel Prize. And I think the great example of this is Dr. Martin Chalfie, who won physiology or medicine in 2008, working on green fluorescent protein. So if you work in immunohistochemistry, if you work with staining or anything, you'll know that green fluorescent protein is a very important part of now what we do in biomedicine. It's a marker that we can use for staining to visualize things that we otherwise wouldn't be able to see under a microscope. But his talk actually was titled "Tickling Worms: Surprises in Basic Research," and his Nobel Prize-winning work was actually a collaboration he did with 2 other labs that needed his expertise and his insight. It's funny because his real passion is C. elegans and nematode and studying sensory biomechanics. It just so happened that he did this one side project this one time and accidentally won a Nobel Prize. And he showed a great slide, I wish I had a picture of it, where it showed all of his publications, all of his great works and prizes he won. There were like hundreds and hundreds of, well, not hundreds and hundreds, but there were hundreds of little things on his chart. And there was one little point on the far right that was in a different color. And he was like, oh yeah, that one won the Nobel Prize. So the key with, I think, any success in lab work or graduate study work, and this is something that I suffer from a lot, is to not put blinders on and to really spread out and study things that you might not think are related to your work necessarily, but maybe tangentially, and maybe you'll find some sort of relationship there. This is an image I took of a neuron that's expressing green fluorescent protein. So I have to actually directly thank him for his contribution to science, so that I was able to take this picture. It's not a great picture, but okay. Number 4, never stop learning and never stop teaching. Share, collaborate, and talk. And I think the purpose of this week-long meeting can be distilled down to this idea of collaboration, connecting with one another and educating each other. And Bert Sackman, he won in 1991 for the study. We actually shared the prize with Erwin Nair on the function of single ion channels in cells and attachment. technique. And if any of you are neuroscientists or neuroengineers, you'll recognize his work. And I think he was a perfect example of leading by, leading by example. And in particular, he led a discussion group that I was fortunate enough to attend. The discussion groups in the afternoon sometimes were full of 100, 150 students, and his discussion group was maybe 20 of us. And what was really nice is that he sat and we talked for about an hour and a half, and he went down the line and asked us all about our research, asked us how it was relevant to medicine, and then he gave his own insight into the research that we're doing and where we should be looking to further our studies. And I was really just inspired by the fact that this Nobel Prize winner, someone whose work I admire and whose work informs my work now, years later, would be willing to sort of sit and talk with a bunch of students. And I think that's the key, whether you're a grad student or whether you're a postdoc or you're a PI or you're a acclaimed Nobel Prize winner, talking with others, sharing your excitement and enthusiasm for your work is a key part of being in science or in academia. So number 5, inspiration can come from anywhere. And the subtext of this is that you really can't force science, and you really can't force your studies. I'd like to bring up this Nobel laureate who won in physiology or medicine in 2007. And his talk was titled, Where Do Ideas Come From? And I really wanted to stress his presentation. So he was having issues, and I don't know if you can read this, but this is actually from his lab notebook in 1954, October 18th. And this is the great thing about his talk. He actually brought his lab notebooks with him. These really old, yellowed notebooks that were framed, and he showed us his handwritten notes. So with gel electrophoresis back in the 50s, the issue is that if you ran, so in gel electrophoresis, you can load DNA into it, and based on the size of the DNA, you can separate out your bands of DNA along the gel based on their size. In the 1950s, this was nearly impossible because you wouldn't get separate, discrete bands. You would just get a smear of DNA. And he had and you can see in his notebook here, he just had issues. This is from 1954, March. I don't know if you can see clearly, but there are some bad words in here too, where he's like, I just can't do it. It doesn't make any sense to me. And he got inspiration for his work. Because you remember when he was a child, his mom would put starch on his father's shirt collars, to make them nice and rigid. And he said, well, we're using starch here. Maybe the idea is that we should melt the starch a bit more. We should put the starch in a different form. And actually, once he was able to do that, he got very discrete, nice bands. And so for him, the inspiration didn't come from sitting in the lab and pounding his head and saying, why doesn't this work? Let me read tons and tons of more papers. Let me read as many textbooks as I can get my hands on. It really came down to just sort of inspiration, a flash in the pan. And he was saying that those moments come more often than you think. They're actually there all the time. You just have to be open to receive them. And I think that's a very good lesson for a lot of us who are starting out or in the middle of our graduate work. We don't really see the end in sight just yet, and we're thinking, when is this going to be done? But I think instead of that, we should be thinking, what else can I learn while I'm in this point in my career? He's actually also an aviation enthusiast at the age of 82 or something. Okay, the point 6 is the penultimate point. Don't be afraid of your data, especially when analysis will take longer than your experiment. And I'd like to bring up Brian Schmidt, who won in 2011 in physics for his work in dark energy and acceleration of the universe. He gave a talk it was part of a panel discussion on big data. And he talked about how in astronomy and physics, collecting data is easy. The analysis is much more frustrating. And he was talking not about gigabytes of data, but petabytes of data per week. So he's getting tons and tons of data, and it can be pretty alarming when you're sitting in front of a computer and all you see are just the ones and zeros flash by and you don't know what to do with it. And I know I suffer from this a lot too, where I'll do all my experiments and I'll put them aside and then I'll take like a week off and, tell myself I'll do the analysis. But what I'm really doing is procrastinating because I'm afraid of what I'm about to see. So I think with Dr. Schmidt, I was able to get a picture with him. His point was that you really have to address the data. Your experiments are great, they're fine, but it's really the data that's going to help you. And I think the big important lesson in science and academia is that, yes, it's great to have good experimental methods. Yes, it's good to do it and look at all that, but you really need to be able to analyze your data and to not be afraid of your data. On a side note, on a personal note, before I was an MD PhD candidate here, as an undergrad, I was interested in astrophysics. And I did a summer internship at Arecibo Observatory, the world's think now, or was at the time, the world's largest radio telescope in the world. And I was convinced I was going to be an astrophysicist, and I was convinced that I was going to help, unlock the mysteries of the universe. And this is me with the telescope. And then in 2007, I said, no, I'm going to go into medicine now, or go pre-med. And I thought I was done with that world, but then I was able to meet Dr. Schmidt, whose paper I remember reading in sophomore year of college, and thinking, wow, this would be great if I could ever meet the guy, but I finally got the chance years later. So the last lesson I thought was important was that we have to think about the implications of our work outside of science or outside of academia. And this is what I like to call the Nobel Dilemma. Alfred Nobel, when he created dynamite, he was created as a businessman, he created it as a chemist, as someone who invented things, who had an insatiable thirst for knowledge. He didn't think that dynamite would be used in the horrific ways that it would eventually be used in his lifetime. And I'd like to bring up this woman, Dr. Blackburn, who won in 2009 for her work on telomeres and telomerase. Her study looks at repeating nucleotide sequences at the ends of chromosomes. And there seems to be some correlation with the length of those repeated nucleotide sequences with aging. So as people age, as they go under chronic stress, those sequences get shorter and shorter and shorter. And so her work has broad-ranging implications into aging, into societal issues along with aging. And what I thought was really fascinating about her talk and her discussion groups was that she spent a lot of time talking about the biochemistry of her work, the genetics of her work. But actually, the really interesting parts of her conversations came when she talked about the social implications of her work. What it meant if we can figure out how long someone might live, or what it means to say that someone has biologically worse age than someone else, even though they might be physically the same age. And a lot of her work now also looks at stress and mindfulness in the body and how that affects your day-to-day life. So I think a lot of the work that we have to do as in academia, in science, is to take a look at our work outside of that context and place it in the bigger context of the outside world. And so throughout the whole week, it was a great time. I met a lot of people, made a lot of collaborations, met a lot of friends, people that I would never ever meet in my life. And I would highly recommend that if you ever get an opportunity to go or to attend, to take full advantage, don't sleep, stay up, talk to people, go to every meeting, and above all, above everything else, try the ice cream, because it's fantastic. I think I had every single one of those. I think I single-handedly got rid of like that. And it's like €1, you can't not do it. And they're everywhere. Anyway, so I'd like to thank Office of Graduate Studies, the Fellowships Office, the Laureate meeting, the Alcoa Foundation who sponsored my trip, as well as my PI, and everyone who gave me the opportunity to attend. And thank you for letting me speak to you about this.
Just hang on to them for now, and then we're going to bring up our next speaker, who is a second-time Books and Bagels speaker now. So he has much more to share with you. And this is Gaurav Goyal, and he's a PhD candidate in biomedical science in the School of Biomed. And he's going to talk today about biology at nanoscale. Why don't you come on?
Thanks, yeah, I was here in the very first Books and Bagels. I was the first speaker Books and Bagels had. So it's good to be back and talk about it. My talk may not be as interesting as John's, but I'll try to make it. So I'll talk about biology and nanoscale. It's more concerning the kind of research I do here. I was told to talk a little bit about myself, so it all started back in India. I was born in India, I'm Indian. In India I was trained to study biotechnology, so I did study all the DNA and biochemistry, molecular biology and this kind of stuff. And from there I went to South Korea. I studied at Giszter, Neuroengineering, electrical circuits, stuff like that. My boss was an electrical engineer, so initially it was a shock for me, and suddenly all the biology I had studied was thrown out of the window. I had to learn new, but it was an interesting experience. In addition to studying about neurons, neural circuits, electrical engineering, I also started flying in the air. My boss could do all this Korean magic. We'll talk about it. And then I came down to Philadelphia, primarily because of my wife. So I'm over here, and here now I work in nanotechnology, micro and nanotechnology. This is not me working in the lab. This image from Google Images, and it is a little overemphasized. I do not work like that, but you get the idea. So I'll give you this little cartoon to read through. So a guy says, well, I need a sexy project to boost up my career. The other, and he says, and to get a better job, and maybe I should work on nanotechnology stem cell for fighting terrorists. Well, I do work in nanotechnology, but I do not do nanotechnology stem cells for fighting terrorists. But I wish if I could name my project like that, I could get a job. That would be wonderful. So the thing is, I don't know how people perceive nanotechnology, but I would say it is not just the buzzword. There is some substance to it. At the same time, it does not have all the answers. So if people say, well, nanotechnology, stem cell, you could do anything, that's not the way to go. But still, there's a lot which you can do with nanotechnology. I want to take a little pause and help you appreciate the phenomena at nanoscale at this point. So let me go to this website here, which has a small animation. So this is the length scale, human, etc., etc. 10 is the length scale. So if I start to go down in it, you see mice, you see eggs, you see common earthworms, ants. Then this brown thingy is the human hair. This is all that you can see with your eye, but we still go further down into the cells and chromosomes. At this point, you hit the limit of optical microscope. So till this point, you can see things through the traditional microscope as you all have been trained to see. But there's life beyond this scale. So we go further into the cell, and there are these viruses which you cannot see with optical microscope, but again, there are only limited means to see these things and analyze these things. Okay, we still go further, and we reach this DNA and protein. So this DNA is about 2.5 nanometer thick, and it's so hard to access this DNA or other objects at that scale. And because it's hard to access DNA and phospholipid bilayer, it is hard to analyze them or play with them, manipulate them. So this is about the scale at which I operate. Well, you can go further. It's an interesting animation. I could also have taken you in the other direction where you could see like dinosaurs, Eiffel Tower, Earth, etc. But going back to the presentation. So now we can understand that biology exists at nanoscale. So DNA, proteins, antibodies, phospholipid bilayers, and everything. When we talk about medicine, we see anything wrong with these things, it manifests at the macro scale. We see organs going bad, we see bones going bad, we see some disease happening, but everything actually starts at this scale. And maybe one of the ideas is to be able to understand everything, to be able to fix everything, you somehow, somewhere need to access to these macromolecules and do something about them. And so what nanotechnology work I do is I try to develop means to somehow look into them. And I do that at single molecule level, so I'll talk about that. So if somebody asks me in one word what I work on, then I say I work on nanoscale resistive pulse sensors. These guys are sensors, they primarily constitute a very thin membrane which is insulating. I make a small hole in them, and I fill salt solution on either side of them. So this is the only way for the current to pass through in this circuit. And I put electrical potential against this membrane. So with electrical potential, there is always ions going through, which result in some ionic current. And if there is something present in the salt solution and it also passes through this pore, it somehow prevents some of the ions from passing through, and this transient phase of passing of this particle results in a small drop in the ionic current. Based on the size of this particle, the shape of this pulse may change. So whether it is small or big or long or fat or stuff like that. So we fabricate these membranes, these pores, we run these kind of experiments, and we get this data and we analyze and try to make sense. How I do it is, now this thing is only 50 nanometer thick, so I gave you an idea of how much a 50 nanometer could be. If I stack hundreds of 50 nanometers together, I might reach to one human hair. So I have to make it really, really thin. And then these pore sizes are even less than 50 nanometers. So here's TEM video. So I take my samples in TEM, which is a transmission electron microscope, and I use the electron beam to knock off some of the atoms from this membrane and create this small hole. On the right, you have the hole that I made in graphene, which is single layer carbon, and I can make these holes, and I can also shrink these holes down. The scale bar here is only 2 nanometer, which is the thickness of DNA. So the technique gives us the control on the scale of the devices that we create. So now I'm creating something artificially which commensurates the size of the molecules I want to analyze. So I take that chip, I put it in a flow cell, and like this membrane, it has a small pore. I put some protein molecules, I patch clamp it, and these proteins pass through this pore, and this result in these kind of current drops, and we measure the amount of time a protein is spending inside the pore and the amount, the magnitude of current drop. So that is how we correlate these current signals to the protein that we are analyzing. I've studied several proteins. It's an example of transferrin protein responsible for uptake of iron in the body. So protein is native form, and when you put protein in a high electrical density, so it kind of stretches out because different parts of proteins are put together. And with this we can study the stability and folding characteristics of proteins. So with increasing voltages, the amount of current being dropped in the pore decreases, which in a way highlights to that the protein is being stretched as you pass it through the pore. Based on that, you can infer that how the protein is folding, how it's behaving at different pHs, etc., 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 a lot of geeky stuff. These nanopore sensors are also developed or marketed mainly for DNA sequencing. Let me just, this is a small video from Oxford Nanopore, but the idea is, so these people use this protein pore. So just like the solid state pore that I'm making, it's a protein pore. And what they and they put this in a thin membrane and try to pass DNA through it and sequence it. something comes in, you see a kind of current drop. So this is animation of something what I was talking about. Similarly for DNA, you insert DNA and you try to obtain signal for each base on the DNA and try to read it off. So the current technologies that we use for DNA sequencing, they usually chop DNA down and do some chemistry and this and that to get the sequence out of it. But this thing is like reading textbook. You have the DNA, you take it into the board, and you just read it off. So people are still developing this technology. If it works out, then probably you will have DNA sequencing done in clinics just like you get your blood pressure measured. Okay, other thing I've been working with, this is my work with Dr. Ming Xiao in Biomedical engineering. What we do is we create very thin nanochannels. So generally these nanochannels are like less than 100 nanometer in thickness. So DNA in the solution is like in a globular form, like a jumbled piece of yarn. We somehow linearize it and put it in the nanochannels and try to visualize it. DNA is labeled with sequence-specific tags so that when you see DNA, you can figure out what is the sequence. So this kind of helps in mapping. So this is a cartoon, but this is how it finally looks like. So this is what people call DNA barcode. So each of the dot that you see is one of these labels, which is sequence-specific label on DNA. And then I extract some of this information, do some image processing and Gaussian fitting to it, and I can ultimately locate the position of these tags, and then I capture movies of this DNA motion inside the channels, and then I can see how, even in absence of electric field, how the DNA vibrates. So you can study the mechanics of the DNA. You can study how tags stay in DNA at different location, how is the length DNA varies, and stuff like that. People use this technique for DNA mapping. So sequencing is one thing. Sequencing is when you determine each nucleotide, but you also need a framework to put those nucleotides in place. So this provides a broader scaffold, a broader framework, and then when you do sequencing, you can put them in place. So these are like the landmarks on DNA, which you obtain through this technique. So this is pretty much about what I want to talk about. In summary, this nanotechnology gives us the access to these molecules which you otherwise cannot see, cannot access. You do deal with them in molecular biology, but you deal with them on the broad scale. So something unique about the way I do it is, in conventional sense, when you look at protein solution, when you look at DNA solution, you're looking at thousands and thousands of them. The signals that I get, each signal corresponds to one protein, or each signal corresponds to one DNA. So it gives you access down to the molecular level, to the single molecule, how this DNA is behaving or how this protein is behaving. And I think the understanding that we get from it and the ability to perhaps control it or manipulate it can have far-reaching consequences. I want to acknowledge my lab members, some of my collaborators, and funding sources. Thank you.
Is it my turn now to take over the podium?
Please.
I guess you guys have figured out now that I'm the jack of all trades.
is a moderator. Dr. Lim steps in. All right, now I'm going to start it off, the question and answer session, by asking Gaurav some questions. I want to find out how close he is to winning the Nobel Prize in nanotechnology. So let me ask you the first question. It belongs to John's list of 7. Do you believe in your research?
Yes, I do.
Great. Please check it out. Yes. To question number 1. Have you failed?
Yes, a lot of times.
Can you learn from it?
As a matter of fact, one of the examples that John gave about that previous secretary of energy,
Right.
he bounced from labs to labs, etc. John and I happened to be lab mates in the very first year of my grad school year. Yeah. So I also used to work with neurons and neuroscience, culturing cells and stuff like that. But then one day felt that I should move on to someplace else, and 3 days later I was in a different lab. So yes,
So the answer to the third question is that you haven't been afraid to try something different.
yes. So as I said, I was trained in biotechnology, then I moved to electrical engineering and stuff. So my master's advisor was a EE major, and currently my advisor is mechanical engineer. So I started out with more like biologist, but I think it's fun to learn new things and branch out.
Do you continue learning and do you continue teaching?
I guess, yeah.
You do? Check. Are you afraid of your data?
Yes.
No, you say no. Don't be afraid of it, but you are afraid of it.
At times, yes.
All right. Do you ever consider the implications of your work?
Yes, I do.
You do. 6.5 out of 7. Not bad, right? So Nobel Prize in medicine.
Well, they don't have a Nobel Prize in nanotechnology, unfortunately.
All right, John. What about you? You gave us some suggestions about what it is that we should do in our research. You did not tell us, though, how you got to Lindau. Looking backwards, I know that people like Bagel and I had a role to play in choosing you for that stint in Lindau. But looking back, can you figure out, without any assistance from us, what it is that you did that made us feel that you are the appropriate person to represent Drexel?
I think that you guys had everything to do with it. I think it's about having the right support, whether it's your PI, your lab mates, or people that are willing to work with you, and that you're willing to work, that you can work with to really accomplish your goals in research. And I've been very fortunate to have found a nice lab that fosters that environment. And I would say for anyone that's sort of starting out in grad school right now, or if they're undergrads, even to find something that you really care about, and find people that you know are willing to either mentor you in that, or willing to take you in as sort of a student that can learn from you, or that you can, that is a student that's willing to learn. And I think, like I said, just be willing to learn as much as you can, even if it's not relevant to what you think your future might be. I spent 2 years studying quantum mechanics, and I never touch it anymore, but I mean, I still appreciate that aspect of it. So I would say that, and also be in constant communication with people that inspire you, and that people that are willing to work with you.
Do you all know anything about the Lindau Awards competition that we run every year? You guys didn't get the announcement? Are you interested to find out a little bit more about the competition? Right? The first thing that you have to be is that you can't be too far into your PhD studies. So if you've been in the PhD program for more than 3 years or more, you're out. because the selection process essentially targets those who are beginning their graduate studies or those who have been in it for just a year or two. You have to be in the right area because the competition changes from medicine to physics to chemistry and yes, economics. Oh no, but economics apparently runs everything.
Oh, okay.
And as it happened, we were invited about 7 years ago. I can't remember why it is, but it may have something to do with the fact that the person who runs the competition for American nominees happens to be at the University of, well, he's not actually at the University of